or good afternoon, whatever time of the day it is for you. It's good to be with you. All right, welcome to our first lecture in the new normal. This is going to be for biology, our biology lecture on, let's see, 14B. We're still talking about plants, and this class is on March 31st, Tuesday, March 31st. Okay, so here we are, having to do this new thing. I'm going to try really hard to get things to you in a timely manner. First of all, a few logistics here is that this PowerPoint will be coming to you over YouTube. It's a YouTube video. Now, the reason I've done that instead of Zoom or with a regular recording of the PowerPoint is that through those other two, I have not been able to play videos. All right, so videos to me are a very important part of the presentation, and I don't want to leave those out. Some of the other teachers are having you go separately and look up those videos. So I've chosen to go this route, and let's see if it works. If we want to get together as a Zoom classroom just so we can have some live interaction, that would be great, but I would prefer to do these lectures recorded. Now let me know if you agree with me on that, if that is something that's going to work out for you. Of course, the lectures recorded is not a problem. You can listen to me here, you can listen to me there, that kind of thing. The big problem is, of course, is going to be the labs, and I really have great optimism that we will be getting back together in the classroom. I'm not sure when or if, but I really do feel very positively about that we'll be back together. If that's the case, you've done all of your major dissections that you're doing on your own, so that's great. We got that out of the way. We're doing plants for the next several weeks. And then at the end, we do have some dissections. There are demonstrations that I'll be doing. For instance, we have one big snake, and I'll be dissecting it. I really hope that we get together for that one. And then we have also a bird, and then a rat, at the end. So I'd like to be able to get that together, to get together with y'all to do that. If not, I'm going to have to figure something out, whether it's filming it, or, we'll see. We'll see. Time will tell. The other difficulty will be quizzes. So we are still having quizzes on the modules. I can send those via email to you, and y'all can take a picture of them. There's some sort of, scanning thing, one called Genius Scan. You can scan it up and show it to me that way. Or I could send your parents the key, and they could grade it for me. But let's keep everything the same there. Keep doing your study guides, and we will, and I'll give you credit for those. All right? So what you're going to see now is the PowerPoint. It's going to be just like we have a regular PowerPoint each week. So I'd like to have your book out in front of you. Be ready to take notes in there. And at the end, for this particular class, our lab was going to be looking through the microscope. So I will actually have microscopic pictures up on the screen for you. And you can draw those straight from there as if you were looking through a microscope. And that'll be it for this week. And then as we move along, we will roll with the punches and see how it goes. All right, so I hope everybody is well out there. I'm feeling very well. No sicknesses over here. And I hope everybody stays well. I have full confidence in our God that He's going to pull us through this and that something really great will come out of it. So I've already learned how to have a YouTube channel. How about that? So anyway, keep watching, and you're going to see the rest of the video here. It's kind of nice that you will be able to, because it's a regular old video, you'll be able to pause it whenever you like. Okay, so you can pause it, write some things down if I'm going too fast for you. Or if you wanted to listen to something again, or start it over, or go to the restroom, or go eat lunch, or whatever, we have that ability to do this. All right, y'all? So enjoy, and hopefully I'll see you real soon.
Okay, so I hope the sound is okay for you. It's a little bit tinny over here, but hopefully, you can just manipulate the volume and make that sound okay. Here is our new schedule. We see that on the 31st, we'll be meeting here and looking at plants. Remember that I am using Module 14 a little differently than the rest of the ODA group, and that I have 14B, but really it's all just in 14. So you don't have a lab that says 14B. We're just on 14. I'll be giving you a lot of information today that you won't even have to write down. So there you go. You won't have as much writing today. You'll have a little bit after the leaf things we did. Remember, we talked about leaves. We boiled the purple cabbage, And we saw how the leaves were really green underneath there and that kind of thing. So today we're going to look at stems and roots. And then when we get to module 15, you're going to see here we're going to be talking about the reproductive parts of plants. So we'll be looking at flowers, seeds, and fruits. And then I spend 3 weeks on module 16 because all that great stuff, reptiles, birds, mammals. Once again, your note taking is going to be pretty minimal here. Then we have our human anatomy preview where you have lots and lots of notes for that in your book. You won't be responsible for all of that. And we have our rat dissection that day. I really hope we get to do that. We will have a final exam. It is scheduled here on this date, the 19th, the last day of class. It will be a sit down, multiple choice, similar to our midterm kind of a thing. And I hope we're back together by then, hopefully so. So module 14, we're still looking at botany. Today specifically, we're going to look at roots and stems. So I'm just calling this part 2 of this. And you'll see all of this is in your textbook. There's not much of it in your lab book, but it is in your textbook. So you are responsible for all of this information. So keep that in mind. I've always told you that all plants are green, right? That if they're not green, they're not a plant. Well, here's one I found. Somebody challenged me, one of my students before challenged me, and so I said, well, I will prove it to you. I will look it up, and sure enough, I found a non-green plant. This is called the Indian pipe. It doesn't do any photosynthesis. So you think that all plants have to do photosynthesis. Well, this one doesn't do it. It can't, right? It's not green. It doesn't have any chlorophyll. But it is an angiosperm, okay? It's saprophytic. Do you remember what that means? That means it's going to absorb dead things, right? It's going to break down dead things and absorb those nutrients into itself. So sort of like a fungus. And so it kind of looks like a fungus too. So what's the difference? Why is it a plant? Because of this. It makes a flower. There it is. An angiosperm is a flower. So in that flower, it's going to have the seeds for a new plant. So very interesting. That's the only one I've found. That if y'all want to do more research on that and figure it out, you can. But it's a pretty looking plant there. It's very nice looking. But I wanted to get that out of the way at the beginning here. Okay, we're going to say most plants, like the vast, vast, vast majority of plants, are green. Okay, we're going to divide all of our plants into 2 major groups right here. Now, this is an incredibly fundamental, important part of botany. And we're not really spending that much time on it, but the textbook does spend quite a bit of time on it. All right, so keep that in mind. So we're going to be looking at these things called monocots and dicots. It's the 2 big groups. And these words are short for their real word, which is monocotyledon and dicotyledon. And now you see why we call them monocots and dicots. That is a short for that. All right, flowering plants, also known as angiosperms, can be called the anthophyta. And they're divided into 2 classes. The monocots, which have a single cotyledon, for instance, a corn. So these are the plants, by the way, that are going to make a flower. These are some of the characteristics of monocots. They have parallel veins on their leaves. So if you probably think about when you look at a piece of grass, a blade of grass, if you think about it, its veins go just straight up and down parallel, right? And that's because it's a monocot. They tend to have fibrous roots. So if you pull them up out of the ground, if you've ever had to do some weeding and you pull it up and there's all these roots down in there, that's probably a monocot. And when they have their flowers, their petals are they're either 3 or 6 or combinations of 3 to 6 petals on the flowers. Our dicotyledons have 2 cotyledons, so 2 original leaves that come up. The bean plant is a good one. Most of our vegetables we think of would be that way. Their characteristics include netted veination, so a leaf that has not straight veins, but the veins go out like the palm and things like this. Their roots are more like a taproot. So a taproot is a straight root down. Think of it when you pull a carrot out of the ground. That root on there would be, kind of thick and going tapering down. That's called a taproot. And their petals of their flowers are 5 or 4 on the flowers there. All right, so those are our main differences. I'm going to show you a little schematic here. This compares them. Okay, so you see these are just everything that I just told you. There's something else here, the pollen grain, right? The pollen grain has 1 pore on the monocots over here, and three pores over here on these. Those are pollen grains, very small. And then we're going to see at the end, in fact, our lab we're going to be doing is showing the difference between the stem and the root system of the monocot versus the stem and root system of the dicot. So they're quite different the way they look under the microscope. They carry their vascular bundles differently. Now, do you remember when we talked about the vascular part of a plant? Those are the tubes that carry either water up or nutrients down. We talked about that, right? Xylem goes up and phloem goes down, and those are the vascular bundles that all plants have, that all vascular plants have, the vast majority of them. So looking at these again, we can see how the seed of a monocot is quite different. This is a corn seed right here. It only has the one side right here. It doesn't break in two very easily. So if you had a corn seed, you couldn't really snap it into two as easily. You could do that with a bean seed. So when it starts to germinate right here, you can see one leaf is coming up. Here comes that one leaf. So that first leaf comes out as a single leaf coming out of the ground. All right, monocot. That is the cotyledon is right here. Some people pronounce it cotyledon, but it is cotyledon. And it comes up here, and then quickly the leaves will start to grow here at the top. In contrast, our dicotyledon can be broken into half pretty easily. This is a bean seed right here. We will be looking at seeds in Module 15. And inside the seed, you can see the beginning of the plant right there, the little leaves. And there's two of them. How about that, dicotyledon? And we'll learn next time about how the cotyledons are the food for the young plant. So when we put it in the ground and it starts to germinate, the first thing it'll do is send a root down. And then it'll push up, and we'll see that cotyledon come up. You've probably grown a bean plant before, and you can see that those two, it looks like the sides of the bean. They'll come out just like that. And then guess how many leaves will come out? Those first leaves, there'll be two of them because it is a dicot. So any plant that you see coming out of the ground, look for either the one leaf coming up with parallel veins like a corn, or the two leaves coming up, like many things, squash, things like that, things in your garden, tomatoes. All right, so here are our roots. Remember I said that the monocots tend to have fibrous root systems, and the dicots tend to have taproot systems. So here's what they look like. Fibrous roots. I'm thinking most weeds, right? You pull them up and there's all those roots there. And then taproot, this, I don't know, is that a turnip or something right there? With that root, that root, by the way, is there. It's actually storing stuff, but it also, we're going to look at the function of roots is to absorb water. There's three functions we'll look at. Absorb water and nutrients. Usually under the soil, but not always, okay? So usually they will be completely buried, but now and then you'll find them, for instance, like an onion, that'll be partly out of the soil. They anchor the plant. They keep it in place, all right? They don't need to be, flowing off in a light rainstorm or anything, so they will keep them nice and strong. If you think about large trees, they have a very large root system. Many trees are dicots, so many trees have taproot systems, and it anchors them so they can grow nice and tall and not fall over. And three, they're going to store food. All right, so the storage of food for themselves, right? They're making the food in their leaves. They're making glucose, remember our photosynthesis reaction, and then they're storing it down, and root plants, when we talk about root plants like carrots and beets and things like this, we know that they're storing that sugar that they've made down in that root. And who are they storing it for? For themselves to use during respiration times when they have no sun out at nighttime. But we can utilize that, of course, by pulling those up and eating them ourselves. And once again, you can see our root systems are divided there into the fibrous and the tap. And can you remember which is which? Which one's monocot and which one's dicot? You can actually look at those leaves and tell pretty easily, right? Taproot is the dicot. Usually roots must be quite a bit larger or longer than the actual plant. And you know how that fibrous root, if you added all those together, that would be a very large root system right there. So we do this to increase the surface area. It maximizes the absorption of water. We know how important water is in the photosynthesis equation. And so it's important for the plants to get the water. This is especially true of trees. So they say that if you add up all the little pieces of root in a tree, that it'll be 5 to 10 times longer than the tree is tall. Wow, there's some tall trees out there. So I put this picture up. See if we've added all those root things together right there. In this particular tree, you might guess that that's a fibrous root system, but actually it's a series of taproot systems going on right there. But if we added all those together, it would be 5 to 10 times taller than that little tree right there. You think of some of the really large trees, which we'll get to in a minute in this lecture, and how they can be very, very tall. They must have some deep, deep roots. Looking close up, this is a microscope view of the cross-section of a root, okay? So we're going to look at the 4 regions of the root. So I will point to them here. Right there is the root cap. It's down at the end. Notice those cells. Do you see right above it? So right here, you can see these cells have those little purple dots in there. Remember what that is? Those are the nuclei, right? The nucleus is there. You can see it all the way through here. Look at all those living cells right there. But look at the cap. See, none of those nuclei are present right there. That's because those cells down there are dead. They're like the outer part of our skin, if you will. It's the thickened part. It's tough. It pushes through the soil. Sometimes soils can be very, hard to get through, and so these roots will be very tough at the very end right there. Next comes the meristematic region. It's right in there somewhere. And remember that the word stem, you see it right there, stem. These are stem cells. These are the cells of the root that are deciding if they're going to be, what part are they going to be in the root? Are they going to go down and become the root cap? Are they going to eventually become part of these other regions right here? Then comes the elongation. This is when cells begin to differentiate. And you can see in the word elongation, that's how the root is going to grow tall. So the tallness is growing right in there in the elongation part. And then when they're fully differentiated, you can see those cells right up in here. And notice how these ones in the middle, that's sometimes known as the pith. And the pith oftentimes will have hardened material as well, and not alive anymore. So no nuclei. All right, so you can see all those parts of our root. And let's look at the root hairs. These are fun. If you take some seeds, and you can do this at home if you like, take a few seeds if you have a garden, even if you have just some dry beans that you use, your mom cooks them, take some of those and put them in a wet paper towel, and in a few days they will start to germinate. And when they germinate, the first thing they'll do is they will send out a little root. And this root right here, you can see, has these excellent little projections here called root hairs. And root hairs are there to increase the surface area even more. And why do we want increased surface area? For increased absorption of water, right? And I'm going to show you this, a great picture here coming up. This is very, very close up. At the very beginning of the root coming out, right when it comes out, it already has all of these little root hairs on there to absorb all that water to pump into this. This is the seed coat, by the way, called the testa, and it's broken now. And these are the cotyledons right in there, which will come up, and the leaves will be hidden right down in there somewhere. So the first part to take place in the germination is the root coming out. And it's interesting, which I believe we'll learn in Module 15, the root always knows which way is down. It knows which way is gravity, where gravity is. So that's pretty cool about plants. God has, put that into their DNA. So here we are looking at that cross section again. We're going to see, remember, here's what I showed you, the maturation region, the elongation region, the meristematic region. Remember, these are going to go this way or this way, depending on what they become. Here's a cross section. We're going to be drawing that at the end of this module. We see our xylem and our phloem right here. And then also we're going to be looking at the parts here, like the epidermis would be the outside region here, the epidermis, outer part here. The cortex would be in the middle, like the core. Endodermis, do you see that on there? So our epidermis, our endodermis right in here at the top. It's going to surround the vascular chamber. Remember what the vascular chamber is. That's going to be our xylem and phloem. And our vascular chamber holds that right there. Okay. And you can see it on this cross section of this. This is obviously, I can tell by the root, this is a dicot because of the way that the vascular chambers are arranged. All right. Here's a microscopic view of it. All right, we can see here the xylem. Do you remember from last time? It's been a while, hasn't it, that we've been together, but we talked about the xylem. That tissue is not alive anymore. It's like straws. It's like straws in the plant that are pulling the water up. The phloem is still alive. It's right in here. And that is taking the nutrients from the leaves down to the rest of the plant to feed the rest of the plant, to store the sugars in a root or in a fruit or in the stem, depending on what kind of plant we're looking at. Here's our meristematic tissue right here. Look real close to that xylem and phloem. And some of that might become, xylem and phloem, depending on what the plant needs. So look at the difference between these two. Okay, they're quite different. So you could say, well, they're two different plants. Actually, they're two different types. So one is a monocot and one is a dicot. Do you have any guesses? This might be your biggest hint right in here. When you see that X right there, which I just showed you on the previous slide, when you see that and these bundles are arranged that way, you can be pretty sure that's a dicot right there. The monocots tend to have scattered vascular bundles. All right, so you at the end when you're drawing this stuff, you will get to know that, and when you're reading it in your text as well. There's our dicot root. You see that X in there? X for xylem. That's a pretty good way to remember it. Our phloem right next to it there. And then our xylem is arranged sort of in that circular pattern right there on the monocot roots. More scattered than put together like the dicot. This is a little animation here. It's going to play over and over again, so I'll have time to explain what's going on. It turns out that plants know stuff. Like I told you earlier, they know where down is. So that is called gravitropism. We believe there's a hormone that is part of that deal. So note how it knows to go down. And look at that second one. Okay, let's get back to the second one. It says thigmotropism right there. That's when it can feel something near it and it'll go around it. And then the last one, of course, is hydrotropism. Notice it knows where the water is and it goes for it, right? It reaches for it. So one of the fun things about that thigmotropism is it also is not just in roots, it's also in stems. If you've ever grown a plant that grows up a trellis, when the stem hits the trellis, it feels it and it will actually start to grow around it. And that's why they grow up a trellis, because they are a type of plant that has a hormone that gives them the ability to do that. All right, moving on to stems. They have three functions. They support and manufacture the leaves. All right, so they're holding onto the leaves, which we know are important because that's where the food is made. They carry water and nutrients to and from the leaves. And some of them will actually perform photosynthesis, okay, because there's two types of stems we see over here to the right. The herbaceous type is this green one over here. And remember, if the stem is green, the stem can actually perform photosynthesis. But check this one out. This is a woody stem. There's no photosynthesis in this stem. This would be getting photosynthesis only from the leaves of this plant. This looks like a tree branch or something. This almost looks like a fruit tree stem coming up there. And so you probably noticed that if you have a garden, that a lot of your garden plants will have green stems, right? Because they're usually going to have quick growth and this kind of thing. Whereas things that take longer to grow, bushes and trees and things, will usually have a woody stem. So here's our monocot versus dicot stem. Look at the big difference here. There was quite a bit of difference in the root, but the stems will be very, very different. You can see our vascular bundles are just completely scattered over here in our monocot. There's a group there and a group there, like that. When it comes time to draw these, we're going to get a close-up of that. They kind of look like a little face yelling at you. Maybe a monkey or something. And then over here, our vascular bundles are kind of arranged in this circle right here. Remember, this is the stem now that we're talking about. In the middle, we have pith. Pith is tissue that's no longer alive. It is giving structure to the plant. It's getting stability to the plant. Remember, a stem's going to get them nice and tall, this kind of thing. You can see our phloem, right? Is it the light blue right here? And the xylem, those holes right there. And we know all the traveling going through there. And I'm not sure what this plant is supposed to be. Our epidermis out here. Yeah. So this is the monocot stem. And here's what I told you, these little things that kind of look like a little face looking at you. and it shows the phloem and the xylem surrounded. This is the bundle, right? So the bundle sheath, we talk about a vascular bundle in the stem of a monocot. And there's those bundles. Here you can see it almost looks like a little alien face looking at you, right? It's so kind of creepy right there. We have xylem and phloem in the bundles. Here's a strange looking one. Yeah, so we'll see some of that in our lab. Herbaceous stems, these are the green stems. They don't grow thicker, they only grow taller. They're green. They can perform photosynthesis. Sometimes they're hollow. I know with the zucchini plant, if you cut the stem off, you'll see it's just a big hole in the middle there. That's that pith area. Some of them, though, will have actual cells in there, often dead cells in the middle there of that pith. You can see our vascular bundles all around the edges, just like we saw in the previous slide there. Our woody stems, does this resemble anything in your mind? You think of something right there? It looks like a tree, like the middle of a tree, right? So a tree would definitely be a woody stem, wouldn't it? The woody stems do not perform photosynthesis. They grow thicker. They have special cells in here called the vascular cambium. So this is the living stuff right over here. This is the vascular cambium. In the middle, we have the pith. Also, here it's called the heartwood often, and that is usually just for strength and everything, not living anymore. We have all of our vascular tissues around the edges here. So if you're looking at a tree, you know, on the edges near the bark is where the xylem and phloem are. So the meristematic tissue continues to make xylem and phloem, adding layer upon layer. So they're solid all the way through, and those layers can be seen year after year, right? They lay down what we call tree rings. These rings are formed that way. Pretty cool. There's also types of stems called subterranean stems. You might have thought that a potato was a root that you were eating, but it's not a root. It's actually a stem. That's a botanical distinction here. And it comes, is where they are storing the food, the starch that they're making in the leaves. They're storing them down here in these subterranean stems. And they're very starchy. We know that. We can dig those up and use those for food. Each one of these are pretty cool. These we call the eye. Look in the eyebrow. That little eye right there is capable of growing a brand new potato. So if you had a potato and you cut it into pieces, and each piece that you had an eye on it, at least one eye on it, then those would grow new potatoes. Cactus plants also have interesting stems. Okay, you might have thought that we're looking, you know, at a trunk there, but it is kind of a trunk. It's a stem. So where in the world are the leaves of the cactus? So these stems actually hold a lot of water in them. I'm gonna give you the next picture right here. So here is the stem of the plant, this fleshy part, and see the needles. What do you call those? The spikes right there. Those are actually the leaves. All right, so the leaves of this plant. Where do you think most of the photosynthesis is taking place in this plant? Probably in those stems, right? In the green part right there. Yeah, pretty cool. Trees are one of the most commercially important plants on Earth. They're dicots for the most part. We do have banana trees and palm trees that aren't even trees, honestly. They're actually overgrown grasses. They're monocots. They have those, think about the leaves of a banana or a palm tree. They have those straight, you know, parallel veins on them. They have woody stems. Those are the trunks. They provide us with so many things, including food. We get nuts and fruit and things from fruit trees and nut trees. They provide shelter. They are wood for our homes, that kind of thing, furniture, artwork. They make oxygen, of course. They protect animals who hide in them. There's just so many things that trees are good for. I don't know that your book talks a lot about trees, but I think trees are amazing, especially this time of year when they're all leafing out with their beautiful green leaves and going through the cycle of life here in the spring. Just beautiful. So the tallest living tree is 369 feet tall. It is a redwood in California. If anybody has been blessed to go out to California and see these redwoods, they are a sight to behold. They are massive. You know, you look up to the top and you can't even see the top. It's so tall. Also, we know that the oldest living tree is a bristlecone pine called Methuselah. in California. It's about 5,000 years old. Now, you know how we determine the age of a tree, right? So the best way to determine the age of a tree is by counting the tree rings. Okay. Oh, wait, there's a banana tree. Not even a tree, right? This is a bristlecone pine. I'm not sure if it is dead. I'm not sure. It's so funny that they protect these in California because what happened was in a California state park, one of the rangers wanted to figure out, he saw these massive trees and he said, wouldn't it be nice if we knew how old these trees were? So why don't we pick one and cut it down and count the tree rings and we can determine how old it is. So this was back in the 1960s. And so they did it. And here is the trunk of the tree. This was the one they cut down called Prometheus. And you can see right over here a little magnifying glass. These trees apparently have very fine tree rings. And it's not even round, right? But apparently you can count these tree rings. And when they did, they discovered that the tree was about 4,900 years old. And it became a scandal because there is, as far as they could tell now, they had just cut down the oldest tree living on the planet. And in fact, the ranger who had come up with this idea, the park ranger, he had to go into hiding for a short amount of time because people were threatening his life because he had just cut down the oldest living tree on the planet Earth. And so they were somewhat horrified at this, event that's going on here. We still have it, of course. It is a scientific, it helped us to learn about something. So I think it was advantageous scientifically. But what they've done now is they have gone and found another bristlecone pine that was similar in size to Prometheus. And they have declared it the oldest tree alive on the planet Earth. They have named it Methuselah, and they won't tell us which one it is. So we don't know what tree Methuselah is, and that's because they don't want somebody who maybe is mentally unbalanced or something to go cut it down and say, yay, I just cut down the oldest tree in the world. And so that was a little bit of a scandal, but it does help us to understand how old trees can get. So when we look at the tree rings, you can see here, you've probably all seen a cut down tree and see those rings. I would have today for our lab brought some tree rings in to take a look at. They grow in three ways. They grow deeper in their roots, they grow taller, up, and they grow wider in their trunks. Each year a new layer is added to the tree. Now it's made up of two parts. It's a dark and a light thing that makes up a ring. So you can see the dark part and the light part. I know my pointer is not great for this kind of thing right here, but look at the white part of the pointer, the arrow, the dark part and the light part right here. And that tells you that it was 1 year. So there was a spring and a fall. So the dark ring that's kind of thinner is the fall growth, and the wide white one is the spring growth, because in spring there's going to be a lot of growth right there. So we can keep time, right? We can count how old the tree is by counting these rings right here. I think I have another one. Okay, the layers right here. The heartwood is the center, it's dead, but it does give strength. It's also known as the pith. It gives strength to the tree just because it's not got the nuclei in the cells. It's not actual living cells. It doesn't mean that it's all dried out or anything. The cells around it keep it moist and keep it preserved. Okay. The sapwood is the young xylem carrying water and storing sugar. In the springtime, this will run. So if you ever tapped a tree, you can get some of that sapwood out. And some people on particular trees will make syrup out of it. So maple trees obviously are the most famous for that. But you could basically, I guess, make syrup from any tree. It's just some trees probably doesn't taste very good, and maple tends to taste really good. The vascular cambium comes next. It's a thin layer. It makes xylem and phloem. So remember those meristematic cells in there? They're going to decide, are they going to become xylem or are they going to become phloem? The inner bark right there, right inside the inner bark is the phloem, and the outer layer is the bark. And the bark gets created, it kind of added on there, and as it starts to stretch, it cracks. Because just like if you had something, like if you think about an insect who cracks out of its exoskeleton, that's cracking, and that's why the trunk of a tree has those cracks in it. And some trees, think about, I don't even know what a birch tree is, the bark can just peel off of it. So it's just, but it is for protection. It's also to keep the moisture inside the tree. If you, one of the ways that the Native Americans in the old days wanted to chop down a tree, they didn't have an axe. So before they had metal tools, they would scrape a circle all the way around the base of the tree. And what that would do was that would scratch into the phloem, and therefore the phloem would not allow the nutrients to get down to the roots. And so eventually the tree would die just from a ring around the lower part of the bark. So it's important to not, like if you're using a weed eater or something around a tree, don't cut too deeply into the bark because you'll wreck it. But these Native Americans would leave it like that, and after a couple of years, it did take patience. They could then push the tree over because it would have died by that point. Here's a close-up of the tree rings. So we see the pith, the heartwood, the sapwood, the cambium, and the bark. So remember, the cambium has the vascular bundles in there. The phloem would be right there on the inner part of the bark, right inside here, and then the xylem in there. What are bryophytes? So these are kind of strange plants. They are non-vascular. Okay, so we've been talking this whole time about vascular plants. These are ones that have xylem and phloem. They have those tubes in them. They have those straws, if you will, that help to pull the water up and down. Well, bryophytes are a little different. They don't have vascular tissue. So if they don't have vascular tissue, how in the world do they get water up? And how do they get nutrients down? Well, the answer is they don't get very big. So they have to stay somewhat small. These would be ground cover things that can actually sit in puddles of water to get the water to them if they need be. They do have roots and everything. So here's some pictures of some of the bryophytes. We have the hornworts, the liverworts, and the mosses. So a moss is a plant. We have lots of moss around here, right? So you can see lots of those. But they never get really, really big. They can't get tall. They can't get tall because they don't have any vascular tissue to give them that height. So they stay very small. They oftentimes will also live in conjunction with other plants where they can get, for instance, some of them will grow on the trunk of a tree because in those crags of the trunk, water can collect and they can sit there and get the water from those little puddles forming inside the tree. They also experience alternation of generations. Now this you should be familiar with. If you remember, we studied that when we were looking at some of the fungi. Right? So the fungi go through the alternation of generation, which essentially means that they have the sexual reproduction and asexual reproduction all in a cycle. So they go from a seed to a gametophyte to a seed, that kind of thing. All right, so like I said, they have no vascular tissue. They send out little shoots and little rhizoids, which are just like, they'll hold on to keep them anchored down. Here is a picture of the alternation of generations. So this is a cycle that goes on. We have our gametophyte generation, which is haploid. That means half. All right, so this is the sexual reproduction part right here because they only have half and they need the other half in order to make a complete organism. Down here, we have our diploid sporophyte generation. And you can see that we're going to make we're going to go through meiosis over here. And we're going to actually watch a video on this in just a minute, but we're going to look at it with ferns in particular. So there's some mosses. You can see it down on the ground, growing really close to the ground, all right, in a forest, which would have, probably some good moisture going on there. This particular kind of moss is important in the ecosystem because this critter right here, the reindeer, survives in the winter by digging under the snow and finding those mosses and eating them. And that's how he survives through the winter because there's no, most of the trees have dropped their leaves. Some of the conifers still have their leaves on them because they're evergreen, this kind of thing. But this little guy can sniff them out through the snow and eats them. All right, so now we're moving to our seedless vascular plants. This is where we're going to see, once again, a alternation of generation in this particular group. These are the tracheophytes. They're seedless plants with roots. And we have one in particular that you should know called a fern. They're in the phylum Pterophyta. They don't produce seeds. All right, so they're seedless. They do have vascular tissue, so they do have xylem and phloem, so they can get large, but they don't make any seeds. So how in the world do they reproduce? Well, they go through an alternation of generations like the mosses do, like the bryophytes, and have this kind of fancy deal. They'll end up with something called a spore, so which is kind of like a seed, but different. And I believe we'll probably talk about that in Module 15. Here is the alternation of generation. We have our meiosis, producing haploids. We have our mitosis, that will bring us to the diploid zygote over here. And this guy that I'm going to show you in this video next, he's very enthusiastic about these ferns. So it's an interesting video, but he gets some really, really great information. All right, so give him a chance.
Ferns have alternation of generations because you can find two unique multicellular body types that ferns have. Ready for some more words? Okay. The two kinds of bodies that ferns can have are either going to be a sporophyte or a gametophyte. Sporophytes make spores, gametophytes make gametes. What do you know? There are some fern sporophytes behind me. Let me show you them. So this is a fern sporophyte. Sporophytes are diploid, meaning they've got two copies of the genetic information. These are the leaves of the sporophyte. This is what you typically think of when you think of a fern. And down here, you can see the spores. Ferns have a number of different ways of growing their spores. In this species, what you can see is that the spores are all located on the bottom 4 fronds of the fern. So you've got this one stem, and it's got a lot of different fronds, and 4 of them have been devoted completely to spore manufacturing, right? So here, all those little dots are spores capable of making a new fern, a fern gametophyte, a new kind of fern body. Other ferns will have the spores growing on the underside of each of these fronds rather than dedicating entire fronds to spores. It depends on the species. Okay, so that was a brief tour of a sporophyte. Here's another sporophyte in my hair. To reiterate, the sporophyte is the body of what we typically think of as the fern, and the sporophyte makes spores. Sporophytes are diploid, and in order to make spores, they undergo a process called meiosis. In meiosis, diploid cells cut their genes in half, producing haploid cells, and those cells develop into spores. The next step in this story is what happens to the spores. So the spores get released, and they fall on the ground, and they develop into a gametophyte, a creature that makes gametes. So the gametophytes are really, really tiny. I'm going to put a picture on the screen because I won't be able to find one on the forest floor. They're about the size of your thumbnail. But, that's still like a multicellular creature. You can see it with the naked eye. It's got a fully developed body. It's not just a single cell. So the gametophyte is this little haploid plant that grows on the forest floor. Haploid fern plant. It's a fern. It just doesn't look anything like a fern. And it grows on the forest floor, and it releases gametes, eggs and sperm. And when it rains, those eggs and sperm swim through the water droplets, and they find each other, and then they fuse. Fertilization. From this fused, fertilized creature grows a new fern, a new sporophyte. So this is what alternation of generations means. It means you've got two distinct body types that alternate from one another. Sporophytes make gametophytes, gametophytes make sporophytes. They're all the same creature. You would call them all ferns. They're the same species. They just have radically different body plans. Some bodies are diploid, some bodies are haploid, and they give rise to one another. This might sound...
Okay, I like that guy's enthusiasm. He's a lot of fun, I think. He really likes his ferns, huh? All right, so we're looking at a fern frond right here, and this happens to be one that is carrying its spores on the underside of the frond, like he mentioned in that one part of the video there. So you can see those little black dots on there. Those are the spores, and they will release those when the environment is really ripe for them. So there needs to be enough water, this kind of thing. You know how if you've ever tried to raise a fern, you have to like spray them frequently with a with a mister or something like this to keep them nice and wet, but not overly wet. I of course can never keep them alive. That's just me. Don't have a green thumb. But when the time is right, they will actually drop those spores down and new ferns will grow. Here is another group of spores being formed on the underside of a leaf. Those are kind of interesting looking. Look, little eggs, don't they? So the big difference between spores and seeds is that the spore doesn't have the food to feed the little embryo while it's, germinating. And so therefore they need to have everything in the environment ready to go. They need to have a nice moist environment. and warmth and this kind of thing to grow. This is, if we're going to label the frond, the whole thing is the frond. And you can see that the top part here is called the blade, the lamina right there. And we have the stipe or stipe down here, and then the pinna is the leaflet right here. The mid part's called the rachis. That's similar to what we call a feather, right? And the costa or the midrib here of the pinna. All right, so that's the labeling of a fern. This is a fern frond that's all folded up. You can see sometimes it will curl up like that just for protection, and you can see those little fronds in there. Yeah, beautiful. I think I have another one like that. Here's the fronds curled up, and guess what they have on them? A bunch of the spores right there forming on there. So they probably curled up because it's too wet or too dry. It looks pretty wet in this pretty picture, so they're just using that to protect themselves. So the two phyla for the seed-making plants, we will cover the seed-making plants in Module 15. We'll be looking at the Anthophyta and the Coniferophyta. The Anthophyta are also, that's represented by these flowers over here, also called angiosperms. I guess that's sort of the old-fashioned name for it. Coniferophyta right over here, making a cone. All right, that's a type of flower if you think of it. We'll look at cones next time, I think. So the four phyla we've discussed in Module 14, we have the Bryophyta, which are the mosses, the Pterophyta, which are the ferns, Anthophyta, the flowers, which really we'll talk about in Module 15, and the Coniferophyta, those are the cone plants, which we will also talk about more in Module 15. I think you might need to write this down in your book. There might be a spot for this. There's a spot in my book, but my book's not exactly the same as your book, so hopefully that I'm telling you the truth right there. Which now brings us to our lab. We'll spend the rest of the time on the lab. You can see right here I bought some roots and stems for you to think about. We're going to look at some of these in the scope, of course, but I would have had these for you. We could have eaten them if you wanted. What do we have there? Do you recognize all that? Of course, you see our roots are going to be, this is a beet. Beets are wonderful. I don't know how you feel about beets, but they're delicious. If you do like them, you know that they are sweet. All right, so that plant is storing all sorts of sugar in that beautiful sweet in that root. In fact, some of these beets are so sweet they put so much sugar in there that there is a manufacturer of sugar called beet sugar or beetroot sugar. So they make sugar from that. Then we have our carrot. Look at that taproot right there, taproot that was growing down. If you look closely, you can see little hairs coming off of it, little root hairs. It's got a little twisting there. It has some thigmotropism going on. Maybe a rock was right there when it was growing and it just grew around it. And then we have our stems right here. So can you think of other root plants that we might eat or that we could eat if you were willing? So I can think of like a radish, right, would be one. Is a rutabaga? Is that a root plant? A turnip? Yeah, I'm not big on turnips. I do like beets. And then we have stems. So lots of other stems. Can you think of other stem plants that we eat? Notice the leaves on the top of that right there. The stem plants would be, for instance, one of my favorites, asparagus. We eat the stem on that one. And let's see, can y'all think of something else? Maybe some Swiss chard. We eat the leaf and the stem on that. Yeah, so there are a few. I think right here you're going to see, I cut these open. Kind of a weird picture. You can see when I cut them open, look at that solid root of that beet. Look at all that yummy beet yumminess. So I'm going to make this for dinner tonight. Some beets, yum. And our carrot, nice and solid. If you were able to see the celery up close, you would see holes in there. Guess what we have there? We have those bundles of vascular tissue. So you can actually see them in the celery. In fact, I would have brought a piece of celery that if you take a piece of celery and you cut it a nice fresh cut, and then you put that into the celery into a cup that has some food coloring in it, any kind of food coloring, the water will go up through the tubes just naturally because of the process of capillary action. They'll be pulled up into there, and you can actually get that food color to color the inside of the celery. Then you can cut it and really see those vascular bundles right there. Also, if you leave it in long enough, it'll actually bring the color. So say you put in blue food coloring, you would actually see the blue food coloring in the veins of the leaves at the top of your celery. So give that a try if you happen to have any celery at home. You can do that. You may want to make yourself a salad for lunch today with some carrots and celery and some lettuce and things like that to celebrate these wonderful vegetables that were made with stems and roots. All right, so this is not what I want you to draw yet. I just want to bring this up. We had this slide earlier showing you the difference between our monocot stem with the scattered vascular bundles and our dicot stem with the vascular bundles in a kind of a ring around the outside right here. And we are going to draw 4 things. Is that right? Is that what it said up here? Yeah, we're going to draw. No, it's coming up. Here we go. This is a monocot because it's Zea mays is corn. And this is our stem. You can see those little alien faces there. But instead of drawing this one, I think I want you to draw the next one. So we're going to draw 1, 2, 3, 4 things. Now, where are you going to draw them is a good question. If you can't find a spot in your book, I have a big blank bottom of the page on my book, but it may not be the same with yours. So we're going to draw a stem and a root from Zea mays. Those are corn, so those are monocots. And we're going to draw the stem and the root of some dicots. Ranunculus, maybe. I was having trouble finding the ranunculus online. All right, so here's what I want you to draw for the corn stem. This is a monocot stem. So there you can see that little face right there. Okay, so you're going to draw that. Notice we know what the magnification is. It's 400x. And furthermore, look, it's all labeled for you. How nice is that? And keep in mind, right now you can pause the video and you can have all the time you want to draw that lovely little part there. It's going to show you all of the xylem and the phloem and everything that's in there. Okay, so that's the first thing you're going to draw. Next, you're going to draw the root cross section of the corn. Okay, that's 100x right there. So make sure and write that down. That's always important for me to know. And you can draw this just in a square. You don't have to draw it in a circle like we normally do because we're not looking through the scope right now. You're just looking at this picture. And you can see that it's not labeled for you, but in your book you'll be able to find out how to label that. Or you can actually go back in this video and take a look at some of the labels there. All right, remember, pause it and draw that. And here comes our cross section of the ranunculus or the dicot root. All right, very cool. I think, once again, I'm going to not have you draw this part, but this part. Yeah, so that inner part right there, which is the vascular bundle. See that X shape? Let me go back real quick. See that X shape in the middle? This one almost looks a little bit like a Y shape, but the X shape in here. And then you'll go ahead and draw this, and notice that it is also labeled already for you. Very nice. Although I do not have the magnification of this one. Let's go ahead and call that 400. That's probably close to accurate there. And here's our dicot stem. It might be ranunculus, it might not be. It is labeled for you as well. So go ahead and use this as your picture to draw. Also, don't know the magnification on this one. I'm going to say 100x on that one. And that's the end of our presentation. So remember, you can go back and watch it as much as you want. I hope it worked out. Y'all make sure and give me a shout out to let me know how it went. Okay, I hope it all works out for everyone. And then we will see you again next week. Very good. Thanks, y'all.